Now we are going to study in detail the effect of cofactors. We did discuss in the beginning when we were talking about the basic general features of an enzyme that prosthetic groups are present. So what they are classified totally under is cofactors. Cofactors are considered important because it has been observed that whenever a cofactor is removed from the particular enzyme, that means the proteinaceous part, and the cofactor is removed, that proteinaceous, or when I would simply call it the polypeptide part of the protein, is not able to undergo or undertake rather the catalytic activity which it is supposed to undertake, and when the cofactor is added, then the catalytic activity can take place. Now when we study about cofactors, we need to make sure what are their functions. So they make enzyme catalytically active. The enzyme does not show catalytic activity in absence of cofactors. Okay, so the cofactor, if we have to identify the enzyme keeping in mind the importance of cofactor, what we have is a part known as apoenzyme. Now this apoenzyme is nothing else but the polypeptide which might have undergone structural specialization of secondary or tertiary structure would be formed, or quaternary structure, whatever the structure it takes on takes in the later on stages. This particular polypeptide would be known as apoenzyme, and to this apoenzyme a cofactor which would be added, that totally would be known as enzyme that would be able to conduct the catalytic activity. I hope I am clear that enzyme is not an enzyme only if it is having the proteinaceous part, that is the polypeptide which has undergone the structural complications. It becomes an enzyme if and only if it is having a cofactor along with it. So the enzyme is formed by a component known as apoenzyme, which is a polypeptide, and a cofactor which comes and joins with this apoenzyme. So there would be some sort of bonding between this cofactor and apoenzyme, and depending on how tightly this cofactor is bound to the apoenzyme, we have the types of cofactors. Okay, there are three types of cofactors. Now first thing you have to note that the first one is the prosthetic group. In the beginning we had done a broad classification and understanding that there are prosthetic groups and they are termed as cofactors, but now as we are studying what the cofactors are, what are their types. So first one is the prosthetic group, second one is coenzyme, and you need to remember that both of these are organic compounds. Okay, both the prosthetic group and coenzyme. Now if they are in organic compounds, on what basis they are different? They are different, the difference lies on how well they are attached to the apoenzyme. In case of prosthetic group, the organic compound, whichsoever it might be, would be attached and tightly bound to the apoenzyme part of the enzyme. Alright, the prosthetic group that is tightly attached, it is organic compound tightly attached to the apoenzyme that is known as the prosthetic group. This prosthetic group is responsible for providing the catalytic activity to the apoenzyme. Alright, now for example, we know that peroxidase and catalase are very important compounds in our body. If peroxidase is not working, there would be poisoning inside our body, because hydrogen peroxide is a toxic which is released whenever oxygen is involved in the process of respiration. For the continuous breakdown of that hydrogen hydrogen peroxide, peroxidase has to come to action, and it cannot work if it is not having heme prosthetic group attached to it. Now such is their joining that it cannot be easily removed. They are very tightly bound, and as soon as the catalytic activity takes place, this attachment it goes over, or it can stay attached to the apoenzyme. If it is a prosthetic group, supposedly the apoenzyme is attached to the prosthetic group, then you are bound to keep one thing in your mind, that the association is not going to break down after the catalysis has taken place. Alright, I hope I am clear that because it is tightly bound to the apoenzyme, the association would not break down after the catalytic activity is completed. It would break down in case of coenzyme, because this coenzyme is that organic compound which is attached to the apoenzyme transiently. When I say transiently, that means for a short duration. And what would be that duration? That duration would be in the time of catalysis. All right, when the catalyst has to work, that is when the enzyme has to work, it must have both coenzyme and the apoenzyme, then only catalysis would take place. So in the case of coenzyme, there would be a transient attachment. Now we often say that vitamins are very important part of a balanced diet. If one is not consuming vitamins, the diet is not complete and it is going to be problematic for the overall development of the body, because these vitamins, they work as coenzymes for many apoenzymes. As in the case of NAD and FAD, we talk about niacin and vitamin B12 are very important coenzymes which bind with the apoenzyme, and they are important for various metabolic processes. Only thing you have to remember that why they are different from the prosthetic group, because their attachment to the apoenzyme would be transient. As long as the catalysis is required, they would be attached. As soon as the catalysis is complete, they would go further. The apoenzyme would be left for some other work. Alright. And last one is the metal ions. Again, these metal ions would be transiently attached to the apoenzyme. One thing that you need to keep in your mind is how do these metal ions work? These metal ions, they form coordination bonds. They form coordination bonds with, we take into account Fe2 positive, all those coordination compounds, those P block elements are considered over here, P and D, which form coordination compounds. We do take those particular metal ions, and in these metal ions, they combine with the apoenzyme in such a way that a coordination bond is formed between the active site, and at the same time, they form a coordination bond with the substrate. So they form the bond, they form such an organization, or such a bond is made where coordination bonds with the active site and the substrate would be formed, and this would bring about ES formation, that is enzyme substrate formation. So you get to know that it is the metal ion that brings the substrate close to the enzyme, it brings it close to the active site, and then only the enzyme is able to do its activity, as in the case of zinc. Okay, zinc forms coordination compound with the active site of carboxypeptidase and the required substrate. That would be in our digestive system. We know these carboxypeptidases are responsible for breakdown of glycoproteins. So it would be zinc that would bring both the enzyme and the substrate in close proximity to each other, so that ES complex is formed and the reaction takes place. So all in all, we have three types of cofactors. You get to understand what is the difference between these cofactors, how they are very crucial for the enzyme activity, and a new term is getting introduced over here, that is apoenzyme, and how apoenzyme and cofactor together form an enzyme. So that is all that is about enzymes that is to be understood. I hope you like this topic of enzymes. Thank you.